What is the fluid who is not a liquid? A gas. A gas. A gas is a fluid. That's why your respiratory system is going to be dealing with gas fluids, while your circulatory system is going to be dealing with liquid fluid, which is your blood. So today we're going to learn that the first thing to understand that this heart is a pump. It pumps the blood. It contracts. When it contracts, it creates a gradient. What kind of gradient does your heart create when it contracts? Yes. Pressure gradient. Excellent. So the heart creates a pressure gradient. A pressure gradient is generated by your heart. Then we move on. How is the heart going to generate pressure? Well, you know your toothpaste. When you squeeze the toothpaste, why does the toothpaste come out on the other end? Why doesn't it go in the opposite direction? Because it goes from an area of a high pressure to an area of a low pressure. That dropper that we had when we looked at the Brownian movement, when we dropped the blue dye, we squeezed to generate pressure so that the fluid can, like when you squeeze a lemon, why did the lemon juice come out of the lemon? Because you increase the pressure. So the heart is going to compress so tightly, and the pressure is going to be so high in the heart, so high in the aorta, which is the closest blood vessel to your heart, and all the blood is going to rush out. All the blood is going to rush out through your aorta to go to your tissue. So for now, have we established what the cardiovascular system is? How is the cardiovascular system different from the circulatory system? The circulatory system, are they the same thing? Is the cardiovascular system and the circulatory system, are they the same thing? They are not the same thing, otherwise they wouldn't have two separate names. If they were the same thing, we would not give them two different names. We don't give nicknames in science. Okay, you can have a nickname in life, but you cannot have a nickname in science. You are one. What is the circulatory system? The circulatory system is the cardiovascular system plus what? What is it missing? Now you have the heart, you have the heart vessels, you have the heart conduits, and what's missing? What's missing? What's missing? What's missing from this? Cardiovascular. Cardio is heart, vascular, conduits, vessels.
It circulates the blood to the rest of the
So what is it circulating? What are we?
Blood.
Blood. So the circulatory system is the cardiovascular system plus blood. If it's empty, when you look at a cadaver that has nothing inside its blood vessels and heart, it only has a cardiovascular system. It has no circulatory system because no blood. So we are pushing blood. We are sending out blood. So the first question, something that's going to come on the exam, is what is the difference between the circulatory system and the cardiovascular system? The cardiovascular system is the heart and the blood vessels. That's a little circle. The bigger circle has on the outside blood. If you join blood with them, now it becomes a circulatory system, not the cardiovascular system. And the cardiovascular system is so reliant on gradient and diffusion. So we diffuse in thermal, when we had the two slabs so hot. That movement of these really hot temperature, the temperature diffused, there was a thermal diffusion from an area of a high thermal gradient to an area of a low thermal gradient. From a really high temperature to a low temperature. Diffusion. From high electricity to low electricity. That's diffusion. From high concentration gradient to low concentration gradient. Diffusion. Pressure. From high pressure to a low, that's why they say blood pressure. To see how good your heart is in generating the pressure. That's why people who have heart disease have a problem generating that pressure. So when the pressure cannot be generated, we cannot squeeze the toothpaste. You can't squeeze the toothpaste. Toothpaste is not coming out. You can't squeeze the heart. Blood is not coming out. If the blood doesn't come out, your tissue is going to suffocate. Your blood is rich in oxygen. Your good blood is rich in oxygen, rich in nutrients for ATP. Your tissue needs oxygen for oxidative phosphorylation, for aerobic respiration. But if there is no blood, or if the heart is not pumping the blood fast enough, the tissue cannot wait. The tissue is going to die. So we need to appreciate how the heart is working like a pump. Now we're going to go and look into this blood. This blood that we are going to learn about, the pump, who's pumping blood, your heart. When we learn about how your blood is pumping, your heart is pumping the blood. What is blood? So let's take a test tube and put in a blood sample in it. Put in a blood sample in the test tube. Then we centrifuge. Centrifuge. If you don't pay attention, you're going to have to go watch this lecture again. Double the work, because the questions are coming right from this lecture. And everything was on the study guide. I'm already going to discuss it and explain it here. So don't do your job twice. Do it once.
What is it?
Centrifuge is a machine in which you would have the ability to rotate. You're going to place the test tube in these circles, and you're going to spin those centrifuge so rapidly at very high speed. What's gonna happen is when you take out this blood, it doesn't just have to be blood, by the way. Any particular fluid that you place in a centrifuge, the centrifuge is gonna spin it so rapidly. When you take it out from the centrifuge, it's not gonna look all red. Like here.
It's gonna stop for right now.
Yeah, very good. Where's the stuff you guys got me today? You see, we have the red chalk came in the right time. You see, we have so that's blood, that's all red. When you spin the all red blood, this experiment is very important. So when you spin this all red blood in a centrifuge, then you look at it, it's not gonna look like that no more. It's gonna look all the way red in the bottom, red in the bottom, red in the bottom. Then there's gonna be a yellow creamy coat. A yellow creamy coat, known as the buffy coat. The buffy coat, or the yellow creamy coat. Yellow creamy coat. And in the bottom, you have your red blood cells, also known as erythrocytes. Also known as, when you separate and remove the red blood cells from your blood, that is known as the hematocrit. Yes, you don't know.
Wouldn't there be like platelets and white blood cells in there too? Yeah.
It's going to be a what?
Like white blood cells.
Yeah, we get into that.
Because isn't the platelets also yellow and creamy?
Yeah, we're going to get to that. We're going to get to that. You jumped the gun. Just wait. We're going to get to that. So on the bottom, I'm going to have my red blood cells, also known as the erythrocytes, and that solution of just red packed blood cells is known as the hematocrit. Now when I move up in the buffy yellow coat, that is when you have your white blood cells. So students have a difficult time remembering that the white blood cells are in the yellow creamy coat, the buffy coat. That's where they are. The white blood cells, white blood cells, these are the ones who perform immune defense. And you would have also, the white blood cells are also known as leukocytes. Leukocytes. While the platelets are known as thrombocytes. So, white blood cells, leukocytes. Platelets, thrombocytes. Red blood cells, erythrocytes. Then at the topmost, you would have a clear color solution. Clear color like water. Clear color solution. And that is your plasma. And the plasma has solutes. It has electrolytes. It has metabolic waste. It has hormones. It has proteins. Pretty much everything. Water. Don't forget, the main component is water. So, at any time, you should be able to tell where is something in your blood. Where is my red blood cells? In the hematocrit. Where are my white blood cells? In the buffy coat. Where are my platelets? They are on the buffy coat. Where is the water? In the plasma. Where are the electrolytes? In the plasma. Where are the solutes? In the plasma. What is the metabolic waste? In the plasma. Hormones, plasma. Sugars, plasma. Lipids, plasma. Everything else in the plasma. So that's why the plasma is the largest compartment. The plasma is about 55% of blood. So that sample that you centrifuged, it was 100% looking red. 100%. That 100%, 55% of it is plasma. 55% is plasma, and 45% is hematocrit, red blood cells, and about less than 1% is the yellow buffy creamy coat. These are the platelets and the white blood cells. And it makes sense they are only 1% because these are generated upon need. They are made when you need them. Like there's no cops around, but when you call them, they'll show up. So the population of LIU now is 90% students, 5% professors, and 2% public safety, and like 1% police. But when you call them, the police will come. So that's why, and the platelets. The platelets, let me finish, then I'll get to you. The platelets, they only show up when you're bleeding. So this is why we call them platelets, because they are like like Band-Aid. They're like Band-Aids. These platelets, they're like Band-Aids. They're like Band-Aids. They prevent blood loss. They prevent hemorrhage. So it doesn't make sense for them to be around if we're not injured. Does it make sense for white blood cells to be a lot when we have no infection? Does it make sense when we have platelets, we have no blood loss? That's why the buffy creamy coat is only 1%, because we don't need it all the time. We need it in special times. Then if somebody who has an infection, now it makes sense. Their buffy coat is not 1%, it's going to go up because now they have an infection. But this is talking about a normal person, a healthy person. That's how your blood is going to be looking like. Yes, Mariah.
I just wanted to know the last three words.
This word?
Oh, the last three.
Okay. Metabolic waste, electrolytes, metabolic waste, hormones, and proteins. Even if I write it and my handwriting is not clear, when I post it for you online, you can go and scroll to that particular segment, and I say it as I write it. So I don't just write down. I say hormone, and then I write blah, 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 blah. Then I write protein, blah, 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 blah. Then you heard me say it, so you will figure it out. Okay, so now we have broken down this blood that we are pumping. So blood, contrary to public belief, is not all red blood cells. People think blood is all red blood cells. No, it's white blood cells, a little bit, but they're still there. There's 55% the rest of the blood, water. When you drink water, where does it go? Does it go to the hematocrit? No, that's red blood cell. Does it go to the Buffy coat? No, that's in your plasma. What about the proteins? All these protein enzymes, albumin, and all of these enzymes that you're going to learn, and the hormones, and the electrolytes, sodium and potassium and calcium, and the solutes and water, and the metabolic waste, carbon dioxide and oxygen. Where are they? Are they in the Buffy coat? No. Only in the Buffy coat is the platelet and the white blood cell. Are they in the erythrocytes? No, because only red blood cells are in the hematocrit. They are in the plasma. That's why the plasma is the biggest compartment. So when they gave you that question from lecture, that I think this was the other section, they were talking about, they said there was an athlete, he was running, and the athlete got, I think, saline, or maybe drank a lot of water, and their blood plasma, is it hypertonic to the red blood cell? Is it hypotonic? So they were telling you that this athlete drank a lot of water. Where is the water going to go? Is it going to go to the red blood cell? Is it going to go to the Buffy coat? It's going to go to the 50. So now it's going to make it like more like 60% now. So now the water increased. There's a lot of water. So when the water increased, what's going to happen? Remember, I told you on the exam, before we begin the exam, I told you in water potential, if you had 9 milliliters of water and 1 milliliter of sucrose, who's the solvent? Who's the solute? The solvent is the big one, the water. I have 9 milliliters of water, H2O, 9 milliliters of water, and I have 1 milliliter of sucrose, sugar. So if I add 1 more milliliter of water, I'm going to increase water potential. So I have tug of war, 9 water molecules, 9 milliliters of water, and 1 sucrose who's pulling against them. If I add more water, I'm going to increase water potential. If I add more sucrose, what's going to happen to the water potential? It's going to decrease. So when that athlete who drank a lot of water, what's going to happen to the water potential in their body, which is another way of saying the plasma fraction, the plasma percentage? What's going to happen to it? It's going to increase. So now when you place that red blood cell, when you place the red blood cell in what kind of solution is that? We added so much water. What does the water do to the solution? It makes it more dilute or less dilute? If you add water to Kool-Aid, is the Kool-Aid going to become more red or less red? Less red. So that's dilute. It's going to dilute it. So now they're going to drink a lot of water, which makes the solution, the plasma around the red blood cells, less osmolar or more osmolar? No. So inside the red blood cell, the osmolarity is 300 milliosmoles. Inside the red blood cell, the osmolarity is 300 milliosmoles. In our body, our plasma is also 300 milliosmoles, because we don't want hyperosmolarity or hypoosmolarity. We want U osmolarity. Remember when we said U osmolarity? Remember we began the course by saying we don't want hypercalcemia or hypocalcemia. We want eucalcemia. We don't want hypertension or hypotension. We want eutension. We don't want hyperglycemia or hypoglycemia. We want euglycemia. Same thing. We don't want hyperosmolarity. We don't want hypoosmolarity. We want eusmolarity. So if I tell you in my blood, my red blood cell inside it is 300 milliosmoles, what do you think the plasma gotta be? 300 milliosmoles. Otherwise, we're not going to be in eusmolarity. But that athlete now, because they drank too much water, water is not going to go inside the red blood cell. It's going to be in the plasma. So do you think it's going to remain 300 or go more or become less? When the athlete drink a lot of water,
More up.
it's going to become 400 millismol? Just answer my question. This solution outside, when you drink water, when he drank a lot of water with his mouth, and the water went inside here, where's the water gonna go? Is it gonna go inside the blood vessel or outside? Outside. It's gonna stay outside. He's drinking the water. The water's gonna stay outside. When it stays outside, now what's gonna happen to the 300? The Kool-Aid, it was red. It's gonna go down. So now it's gonna become 200 because the water potential is gonna go high. The milliosmolar doesn't talk about the water. It's talking about the solution. So the solution on the outside was 300. But when you added water to it, you made it lighter. So it's gonna drop. It's gonna become 200 milliosmol. Then the more water he drinks, the more he's gonna get. So his plasma osmolarity gonna drop to 150 because he drinks so much water. Then the red blood cell, the water is gonna move through osmosis. We said osmosis. Water is gonna move from an area of low solute concentration to an area of a high solute concentration. So what's gonna happen to the red blood cell? It's gonna swell until it's gonna burst. So that's how we're careful with the osmolarity of our blood. Same thing, if you don't drink a lot of water, you get dehydrated, you lose water, then the 300 on the outside here, this 300 is gonna now go up. 400, 500, 600. Then the water inside your red blood cell is gonna flow out. So now your red blood cell is gonna shrink, and you're gonna die. So you always have to have osmotic balance. So now we figured out what this blood is all about. So now let's go back to our heart. So now we figured out that we have this blood that is flowing inside, or it's a fluid. Is blood a fluid or a liquid? Actually, there are four categories. There is a solution, there is a suspension, and there is a colloid, and lastly, there is an emulsion. What's the difference?
Suspension can be filtered, right? And then colloid can't be filtered. But suspension and colloid are both cloudy.
Yeah.
But solution is it can't be filtered because everything's already dissolved, so it's a clear.
Okay.
And then the difference is colloid, whatever it's called.
Colloid.
Colloid is more of a solution than it is suspension, but it is both of them.
Okay. So you describe. So description is not the same thing as identification. What is it? So when I tell you what is a human being and what is a monkey, you say the human being, he can speak and he can, but that's not how we define it. We define it by telling us what is it. What is a solution? A solution is a mixture. It's a mixture. You cannot be water by itself is not a solution. So a solution, the first thing is, so we begin when we want to define something, we begin very wide. Planet Earth, continent North America, country the United States, state New York, the borough Brooklyn, the university NYU. So you keep narrowing down.
Is this all the blood? Like, what is all four of them?
So we're getting to that. Please be patient. We're gonna get to that. Just listen. I'm gonna answer all the questions. When I finish the lecture, you're gonna say, "Oh, you already said that." Just pay attention. So these are all our mixtures. These are mixtures. This is not one on its own. These are mixed up. But how are they mixed up? Yes.
But isn't like suspension used for like medicine for like infants and old people because it has larger particles in it?
Yeah. So now you're talking business. So so all of them, that's a mixture, that's a mixture, that's a mixture, that's a mixture. That's what's in common. Remember I told you if you want to tell the difference between something and the other, begin with the similarity. So they're all mixture. But a mixture of what? A solution is a mixture of water and really tiny particles like electrolytes, like salt, like glucose. That's a solution. That's a solution. But a suspension, still water, but it has huge particles like red blood cell.
That's why it's cloudy, right?
Yeah, the cloudy one is the colloid. We get into the colloid. That one is water and red blood cell. That's a suspension because they are huge, like jello. They're so big. It's like you're dropping like big pieces in the water. That's a suspension. That is not a solution. Then we move into the colloid. Colloid is a mixture of water and protein. That's a special category. Just water and protein. A perfect example of a colloid is your cytoplasm inside the cell. So the cytoplasm is a colloid. The cytoplasm is a colloid. Cytoplasm is a colloid. The cytoplasm is not a suspension. The cytoplasm is not a solution. And a cytoplasm is not a emulsion. So if I ask you on the exam, the cytoplasm is an example of suspension, solution, emulsion, colloid. What is the answer?
Colloid.
It's a colloid. The colloid is white and gel and jelly and cloudy. Cloudy, white, jelly, and cloudy, white, gel. And it's made up of proteins, just proteins and water. If you just have proteins and water, and that is the cytoplasm. Then you have an emulsion. What's an emulsion? You know the oil vinegar from Subway, that dressing? The dressing, the oil vinegar dressing, the one they put on the sandwich, Subway. Yeah, that's an emulsion. Emulsion is when you have a mixture, but you don't have a solid. It's a liquid in a liquid. Liquid dissolved in a liquid, like oil, vinegar. Do you see any solid in these? Both are liquid. So a liquid dissolved in liquid, that's an emulsion. Like the oil vinegar dressing and like the breast milk. When the mommy is breastfeeding the baby and the baby is drinking the milk, that's an emulsion because it's milk and fat. Milk and fat, colostrum. Milk that is white and fat that is yellow. Milk and fat that is yellow, colostrum, fat. And those, so the baby is drinking like a milk dressing. You know the dressing you put in a sandwich. So the baby's digestive system is immature. He cannot digest meat and steak and all of that. So they only give him the emulsion of milk, which is people say breast milk. They think it's just milk. No, it's not just milk. It's milk and fat dissolved in each other. Like the dressing, the dressing from the sandwich, the oil vinegar dressing. Okay. All right. So now the blood is not a solution, is not a colloid, is not an emulsion. What is the blood? What is the blood?
Suspension.
It's a suspension. Very good. Okay. So the heart now, in order for the heart to pump, what time is it? 240? 240? It's not 3:30 yet? Okay.
What is this?
Okay. So, let's make sense out of this model. Does everybody have a model? Please look at this heart. This heart is very interesting. I'm going to have it very close to the camera. This heart is very interesting. The heart is a, what is the heart? The heart, it's an organ. Remember I told you, if you want to define something, You begin very big. You begin very, very big. So the heart is an organ. What is the organ made up of? It's made up of chambers and vessels. So you see these rooms, the rooms we're in, they are chambers, and you know the hallways, we go through them. We go through the hallways. So identify your chambers. Open the door. Open the two bottom doors. Do you see the two chambers? So on the exam, last week on one of my lab practicals, I put a post-it stamp here. And I said, what's the name of this chamber? And some students said, the ventricle. And they only got half credit. Why do you think they only got half a point? Yeah, excellent. We need to put left or right. Okay, we have two. You have two. I have a pain in my ventricle. Well, which one? I lost my hand. Which one? I lost my leg. Which one? The answer was the left ventricle. That's the left ventricle. Okay, so now let's talk about the anatomical position. So Rachel asked a good question. We don't describe things right or left based on you. We describe them based on something known as the anatomical position. The anatomical position. So this anatomical position, okay, so before you guys listen to Professor Idona, you can listen to Professor Kadir first, and then you guys can listen to Professor Idona later. Let me finish my lecture, then we can listen to Idona's lecture. The anatomical position is when the skeleton is looking like this. It's facing you with both palms facing outside. So that is the anatomical position. So facing outward. So now my heart. This side is my left. This side is my right. So now this heart is now in the anatomical position. So now what is this chamber? Is it left or right? Left or right? That is left. Is this? What is that? Is this left or right? Look at my right hand. Clue. That's right. So now that's the anatomical position. But if you look at it like this, oh, that's left. Because now it's not anatomical position. You have to place it in the anatomical position that the skeleton is facing you. The cadaver is facing you. The organ is facing you. And then you orient yourself, you make yourself in the position that the organ is in. So now make sure it's in the anatomical position, not your left, his left.
This way, basically. So basically, if you look at it, and you put your right hand there, it's the opposite side.
Okay, that's extra information. We don't need to know nothing about our hands. Just help us understand our position without.
But in the exam, if I sit here, I need to put it this direction.
Yeah, I need to put it this way.
Then you have to move up then. But you gotta move up. You cannot, if on the exam, if you touch it, if you move it, then you can't see from the front. You just need to orient yourself that this is. So you put it next to you. If you want to know the anatomical position, you put the organ right next to you. So this is my left ventricle. Do you see the left ventricle?
Yes.
The ventricles are the biggest chambers and they are on the bottom. So you have a left chamber, open left chamber open. Next, open right chamber is open. These are the two ones in the bottom. At the top, you have the atria. These are the chambers at the top. And the atria, so two atria at the top and two ventricles on the bottom. They are connected by valves, doors that open. When people get sick, these doors reflux, prolapse, fall apart, and then the blood does not get locked out. It falls right back in. So now there is extra blood for the heart to pump. So that's why the heart gets cardiomegaly. It gets really big because they have to pump extra blood. Okay? Okay, so have you identified left chamber, left, right, left ventricle, right ventricle. Left atria, right atria. And by the way, the left ventricle is going to be bigger than the right ventricle because it's gonna push blood to the whole system. But the right ventricle is only pushing blood to the lung, which is right around the corner. Okay? You need an airplane. The airplane is huge because it's gonna go through continents. But the bicycle is so small because it's taking you right around the corner. So here, your ventricle is going to be as big as where it needs to pump the blood to. But the point is, if you make it to the ventricle, you're going out. You're going out the heart. But if you are in the atria, you're going to get from the atria to the ventricle, eventually outside the heart. Where? We're going to talk about that some more. But for now, you need to know in your model, where is my ventricle? So on my exams, I don't ask like really bad detailed questions. I just ask the general, the big one, the continent, the state, and the country. I don't go down asking about the avenue and the street and just know the begin with really big and then narrow down. These valves that are gateways between the two chambers. On the left, you have your bicuspid valve. It has two cusps. It has two cusps looking like that. Bicuspid valve. That's on the left. On the right, you have tricuspid valve. It has three cusps. This valve is very sensitive. Sometimes it gets calcified. You dump a lot of calcium on them, they can't open up. So now you're trying to squeeze blood through them, but they're locked.
Could you write? Could you write like the tricuspid thing on that?
Okay, so this one is the bicuspid valve. Bicuspid valve.
That's on the left side or not?
That's the left. Now you gotta tell me the anatomical position. The anatomical position, you align yourself like that with it. So now, left. Left.
Oh, okay.
So that's left. Right. You see my right? Okay, that's my right. That's my right. So left, bicuspid valve. Right, tricuspid. Tricuspid valve. Actually, all the valves in the world are tricuspid except the valve between the left and the right chambers. Between the left atria and the left ventricle, they are having a bicuspid valve. They used to call it the mitral valve. The mitral valve, you know the hat of the priest in a church, it used to have two cusps like that. So they used to say, oh, it looks just like the mitral hat of the bishop in the church. So they called it the mitral valve. So the mitral valve, students think that the mitral, it has tr in the middle, mitral. So they think mitral has a TR, that's a tricuspid. No, the tricuspid valve is not the mitral. It is the bicuspid valve who is the mitral. Are we clear? Okay, then these are our chambers separated from one another by these valves. So here you have your left atria, and on the bottom you have your left ventricle. On the top you have the right atria, and on the bottom you have the right ventricle. Okay? Now we're going to the outside the body. So your heart, you have to break it down mentally, and then you try to assemble it in the model. So let's do a very easy demonstration of how our blood flows in the heart. Obviously, yes.
Is the pulmonary artery right away? And then
Yeah, we're getting to that. We are getting to that. If you are patient, we are gonna get to all of that. I promise you. Let's begin with the lung. That's the lung. That's the lung. Okay? And I'm going to have, what atria is that? What atria is that? Is this I have two atrias. What atrias are those on the top? This one, is it the left or the right atria? That's the left atria. Then the other one is the right atria. Then I have on the bottom here my big ventricles. Is it left ventricle or right ventricle? Left ventricle. Here, do I have my right ventricle or do I have my left ventricle?
Right.
That's my right ventricle. Between them, these are this atria and ventricle. We call them a big name known as atrioventricular valves. That's the family. That's the family. Then we name them as the bicuspid. It has two cusps. It has two cusps. That's the bicuspid. The bicuspid valve. And these are three, known as the tricuspid. Tricuspid valve. Okay, but both of them are atrioventricular valves because they are between the atria and the ventricle. But on the left, that's bicuspid. On the right, that's tricuspid. Okay, so now how does the blood flow? The blood that is good, red blood, oxygenated blood. Red blood is good blood. Red blood is good blood, meaning that it's rich in oxygen, is rich in nutrients, and it's a plus, good blood. That's good blood. Going to the left atria, then squeezing through the atrioventricular valve, the bicuspid valve, then going through the left ventricles. Then it's going to get pumped to your tissue. This is your tissue of the body. You see the tissue? Then these are your cells. The tissue is made up of cells. Remember on that first lecture, we said the tissue is made up of cells. Tissue is made up of cells. Tissue is made up of cells. The tissue is, you see these dots are nuclei. The tissue is made up of cells. The tissue is made up of cells. These are my cells with my dot. Okay? So now the blood is going to flow in the atria. You see the blood flowing, red, flowing through the bicuspid valve, filling the ventricle. Then the ventricle is going to squeeze. Then when it squeezes, all the blood is going to go out. Please listen. It's going to go out from my left ventricle into the tissue. So let me make it a little bit better like that. So now this is the bicuspid valve, and this is my left atria. This is my left ventricle, and the left ventricle, the blood is going to flow through it. Then when it contracts, all the blood is going out to my tissue. When it goes out into my tissue, it's going out through the aorta. That is my aorta. Aorta. Yeah, it's on the top. But this is how we understand it. Then we gotta go back. Yeah, this is a conceptual diagram. Your atria is not a box. Your tricuspid valve are not circles. Your ventricle is not a rectangle. This is just to understand. Then we're gonna go in and try to tie everything together. So now the blood, so that's step number one. Good blood comes from the lung, rich in oxygen, rich in nutrients, enter the atria, and that's the second step. Third step, pass through the atrioventricular valve, the bicuspid valve, then pass through the left ventricles. That's step number 4. Then we're gonna pump the blood. Number 5 is going to go to the tissue. Now the tissue is receiving oxygen. The tissue is receiving glucose. The tissue is receiving lipids. The tissue is receiving proteins, receiving solutes, receiving all types of hormones. But you know when you order the food, the takeout, it comes in a nice box, and then you eat it, it's gonna look garbage. So you need to get rid of the garbage. So now we finished with good blood. Now we have bad blue blood. So blue blood is bad blood. Wow, this colored chalks came in the right time. Thanks to Amapola and Penelope and Natalie. They got you guys colored chalks. And I'm not gonna forget how Idona got us those gloves. So you guys are supplying the labs.
Give them extra.
You better take them home so that they don't steal them like they did with the gloves.
Okay, I'm gonna take them in my pocket. I'm gonna keep those in my pocket.
I have a question. So, which side brings blood into the heart and which one puts it away? Would it be like
This is left, right? You see left, left, red, left, red, left area, red blood, left. Yes.
so when the blood's oxygenated, it puts it to the rest of the body, and when it's not oxygenated, it brings.
Okay. We now finished with red. Let me do blue, then you can ask a question. So now we arrive to step number 5, and that is we have delivered oxygen to the tissue. Now the tissue got the takeaway. Ordered Ordered Papa John's. So now Papa John's arrived. So the tissue is gonna eat all the oxygen, the nutrients, the Papa John's. Now it's time to send out the garbage. Blue. So step number 6. All the bad blood, the blue bad blood, is gonna go out from the tissue and return. This is through your vena cava. Your vena cava. You have superior vena cava bringing bad blood from the upper extremities, and inferior vena cava bringing bad blood from the lower extremity. Then the bad blood is going to return to the right ventricle. So this bad blood has a lot of carbon dioxide, negative carbon dioxide. It has low carbon, it has low oxygen. It has low oxygen. And it has a lot of carbon dioxide. It has low oxygen. It has a lot of carbon dioxide. It has metabolic waste. Metabolic waste. It has poison, toxic metabolic waste. Okay? And when the right ventricle contract, all the bad blood is going to go out through the tricuspid valve, and then it's going to end. Oh, no, that's the ventricle. Oh, no. The bad blood is going to return from the tissue through the vena cava to the atria, up here. They're going to go up here to the atria. That's step number 6. Step number 6 is when you go from the tissue to the atria. Then the atria is full of bad blood. So that's step number 7. Then step number 8 is going to flow into the tricuspid valve. Then step number 9, you're going to have your right ventricle full of bad blood. Then step number 10 is when your right ventricle pump the bad blood into the lung. That's number 10. Number 10 is when your right ventricle pump the bad blood into the lung. Then the lung is going to convert bad blood rich in carbon dioxide, metabolic waste. It will shunt the metabolic waste outside, and it will keep remove all the bad blood, and then enrich the new blood with oxygen and convert blue blood into red blood. Then we're going to start the cycle again. So the structures are never going to make sense if we don't understand the cycle. So this cycle here that we drew up here, that is a conceptual diagram to help us understand what happens. Now when we hold our diagram, let's now match this conceptual cycle to our diagram. So the lung is not, I don't have the lungs, but let's say the lungs are up here. So step number 1 is when good blood enters the left atria, enter the left atria through these reds are carrying blood to the heart. If you carry blood to the heart, you are a vein. Doesn't matter what kind of blood. As long as you're going to the heart, that's a vein. So the direction here is to the heart. That's a vein. Okay? And then that's away from the heart. That's an artery because it's going away from the heart to the tissue. That's an artery. We call it the aorta. And this is going from the tissue to the heart. Then that's definitely a vein. Blue vein and all, but it doesn't matter. It's a vein. And this guy is going away from the heart to the lung. That's an artery. So if you're going to the lung, you're pulmonary. If you have anything to do with the lung, you're pulmonary. And if you have anything to do with the rest of the body system, you're systemic. So, blood is going to flow from the lung into the left atria through these vessels carrying the blood towards the heart. These are definitely veins. They're coming from the lung. These are pulmonary veins. Pulmonary veins. Then blood is going to flow from the left atria through those bicuspid valves into the left ventricle, and the left ventricle is going to contract, and it's going to send blood away from the heart through this aorta, which is an artery because it's going away from the heart. Then it's going to go to the tissue. The tissue will use the good blood, send out bad blood away from the tissue to the heart. That's a vein, and it's going to the heart. So this is systemic vein. It has nothing to do with the lung yet. And this ventricle is going to pump blood to the lung. So that's pulmonary. Away from the heart, that's an artery. That's a pulmonary artery. Now, the valves are not only between the two chambers. There is a valve right here. The valve that is going from the ventricle to the aorta. It's a tricuspid valve. We said all of the valves are tricuspid. So this valve right here, these are known as semilunar valves in yellow. The valve that is going to the tissue, from the heart to the tissue, and from the left ventricle to the lung here in yellow. These are known as semilunar valves. These are the pulmonary valves going to the heart, and these are your semilunar valve. These are your aortic valve. This is the aortic valve. Okay? So, this is my step number 1. Blood is right here in the top chamber. Blood is up here on the top chamber, going through this valve, atrioventricular valve, known as the bicuspid valve. You see the bicuspid valve. Then on the bottom you have the left ventricle, and the left ventricle is going to pump blood through the aorta. Where is the aorta? This big one. This big red. So it's going to go through this really big red vessel, and this big red vessel right here is going to send blood to the tissue. Then the tissue is going to use it, send bad blood through your vena cava. These are your vena cava, and we're going to send bad blood to the right atria right here, and through this tricuspid valve you're going to have, through this tricuspid valve, you see my tiny finger going here, that's my tricuspid valve, and the bad blood is going to flow into the right atria, into right ventricle. When it contracts, all the blood is going to flow through this blue region here to go back to the lung. Smells good. Why is that? Okay. So, smells good because the red heart. Okay, anyway. Now, investigate your heart. Have dividing groups and start to discuss the heart. Time to take another chocolate. You like this?
I like it.
Really? Okay. I'm gonna go say he talks a lot. He doesn't do a lot.
No, that's it.
Alright. I'm gonna post this up for you guys. Okay, so this is blue.
What is that? What is this?
That's I mixed
Gatorade with water.
Gatorade with. It's supposed to get lighter if I mixed it with water. It's darker. It's it's a grape. It was Fanta, blue Fanta, blueberry Fanta. You can have another chocolate.
That's like, no. Why are you that? This is like your fourth of the day?
Oh, a lot more.
I hate sugar.
Does your family not have this with diabetes?
They have a lot of diabetes.
And look at you, Professor. You are gonna be the surgeon.
Yeah, and I got a surgery.
For diabetes? What kind of surgery?
This makes me happy.
Yeah, because you want to get another surgery.
What did I want to Is peanut butter. Is Reese's cup of coffee?
Yeah. Just take it over. So.
Is Reese's cup kosher? Is Reese's cup kosher?
Oh, I don't know. If it has a K, if it has a K on it or a U on it, it's
What about
kosher.
it? Yeah.
Is it kosher?
Is there a K or a U on
Why
it?
would I know?
Yeah, it's not.
You asked me a question. Is it kosher?
Check for a K or a U.
Check for a K or a U. Probably not kosher. Oh yeah, kosher.
It is. You can have it.
No, take it. I'll take it.
She said she wanted it.
Take it. I want another one. Do you
You
have
eat
another one?
kosher only. Rachel, you only eat kosher.
Rachel.
Yeah, no bacon.
Rachel, you never had bacon? Wow.
No, you're missing out.
I only have turkey bacon.
I mean, I can't eat bacon.
And beef.
Have you ever tried bacon? Oh.
There's
Yeah,
fake
solid.
Yo, we're so Russian.
I used to hate Reese's cup when I was a little. I
Bro,
was
you put on the piece of bread, bro. Yeah, you put a piece of bread.
It's because it makes me happy. Because me, I have no life.
But something that makes you happy doesn't mean it's healthy for you.
Because I study.
You have no wife?
Yeah.
Wow.